There's so many mysteries of glycoscience that a person could have, 100 careers and still not run out of interesting questions to answer. Well, the motivation for inventing bioorthogonal chemistries really came from frustration. It started with a very specific application in mind. I came out of graduate school as an organic chemist who knew how to make sugar molecules. And then I went to postdoc in a cell biology lab that was interested in studying how cell surface sugars, complex sugars, change in the context of disease. And that's where the frustration hit, because we needed to be able to look at the structures of these sugars on cells in a living system without destroying the cells and chopping things up to bits and pieces. And at the time, and this takes us back to the early 1990s, there really wasn't a technology where one could actually visualize sugars on cells. We couldn't see them. Frustrated by that, when I took my first faculty position at UC Berkeley, the first project I wanted to tackle was develop a technology for imaging cell surface sugars on live cells. And that's where bioorthogonal chemistry came from. There was a very specific application in mind, but once we had the chemistries up and running and saw how powerful they were, all kinds of other applications came to mind, both in my lab and in many other labs. These days, we're very interested in understanding the roles that glycans play in transporting biomolecules across the blood-brain barrier. And that's an area of research that's quite new to my lab, and was launched by a serendipitous discovery of what the industry would call a brain shuttle that was based on glycobiology. People have used bioorthogonal chemistries in material science, in studies of plant biology. So I think, it's as wide as the imagination. I'm really excited about the JACS Symposium. It's an opportunity, first of all, to celebrate with the ACS and with the chemistry community. It's an opportunity for me to meet people that I don't already know and reconnect with old friends and do some networking and maybe even recruit a couple of students to my lab. The students are the future of the chemical sciences. They are the people that, will carry the beacon of light, and build upon the legacy of their predecessors. So the opportunity to meet them and hopefully inspire them and be inspired by them is something I really cherish.